Hello students, today I bring for you a new chapter and that is biotechnology and its application. In this chapter we are going to cover this PDF as well as this NCERT. So let's see the topic what we are going to discuss: biotechnology and applications in agriculture, biotechnical applications in medicine, transgenic animals, ethical issues. So let's start from this PDF about this chapter here. Biopharmaceuticals, therapeutics, diagnostics, genetically modified crops, processed food, bioremediation, waste treatment and energy production. So we use biotechnology in all these different industries. So let's start from the beginning. NCERT says, and this PDF says that three critical resources area of biotechnology. One is to provide best catalyst in the form of improved organism, usually a microbe or pure enzyme, creating optimal conditions through engineering for catalyst to act, and downstream processing technologies to purify proteins or organic compounds. First of all, application in agriculture. Let's see three options for increasing food production. One is the agrochemical based agriculture, second organic agriculture, and third is the genetically engineered crop based agriculture. Here a term given GMO, that is genetically modified organisms. These are plants, bacteria, fungi and animals whose genes are altered by manipulation. Coming to advantages of gene modification in plant. Here it makes crop more tolerant to abiotic stresses, that is cold, drought, salt, heat, etc. Pest resistance crop reduce use of chemical pesticides. It helps to reduce post-harvest losses. It increases efficiency of mineral uses by plants, that is, it prevents yearly exhaustion of soil fertility. It enhances nutritional value of food, example golden rice, which is enriched in vitamin A. GM is used to create tailor-made plants to supply alternative resources, starches, fuels, pharmaceuticals to industries. Next is pest resistance plant. Pest resistance plant act as biopesticides. It reduces the need for insecticides. Example BT cotton, BT corn, rice, tomato, potato, soybeans, etc. Coming to BT cotton, some strains of Bacillus thuringiensis have proteins that kill insects like coleopterans, that are beetles, Lepidopteran, that is tobacco boardworm or armyworm, and dipterans, that is flies and mosquitoes. Bacillus thuringiensis forms a toxic incidental protein, that is called Bt toxin crystals, during a particular phase of their growth. It does not kill the bacillus as it exits in as inactive prototoxin. Here see the diagram. When an insect ingests this inactive toxin, it is converted into active toxin due to the alkaline pH of the gut, which solubilize the crystals. Here Bt, Bacillus thuringiensis, Bt corn, Bt cotton. Here this is the insect who ingests the Bt cotton, and this Bt toxin crystals, they goes inside it and they become solubilized because of the alkaline pH of the gut, and they become activated. Next, the toxin binds to the surface of midgut epithelial cells and create pores. This toxin binds with the surface of the midgut epithelial cells and it creates pores. It causes cell swelling and lysis and finally the death of the insect. Next, Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into crop plants such as cotton. Most Bt toxins are insect group specific. Remember, most Bt toxins are insect group specific. The toxin is coded by a gene, that is CRY-IAC, named CRY. Example proteins encoded by genes CRY-IAC and CRY-IAB controls the cotton bollworm, that of CRY-1AB controls corn borer. Both CRY-1AC and CRY-1AB controls cotton bollworm and CRY-1AB controls corn borer. questions are asked. Another application, nematode resistance in tobacco plant. A nematode, that is Melioidosin incognita, infects the roots of tobacco plant, causing a reduction in yield. It can be prevented by RNA interference, RNAi strategy. RNAi is a method of cellular defense in all eukaryotes. It prevents translation of specific mRNA, that is silencing due to a complementary double-strand RNA molecule. Here you see the source of complementary RNA is formed and infections by RNA viruses or mobile genetic elements, that is transposons, that replicate via an RNA intermediate. Using agrobacterium vector, nematode specific genes is introduced into host plant. It produces both sense and antisense RNA into the host cell. These RNAs are complementary, so they form a double-stranded RNA. It initiates RNA interference and silences the specific mRNA of nematode. Thus, the parasite cannot survive in a transgenic host expressing specific interference of RNA. So, this is all the applications of biotechnology in agriculture. Next, coming to applications in medicine. Before starting this, let's see your NCERT what it says here. The application of biotechnology includes therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. Three critical research areas of biotechnologies are: first, it providing the best catalyst. It providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. It creating optimal condition through engineering for a catalyst to act. And third is the downstream processing technologies to purify the proteins or organic compounds. Let's see. Agriculture, three options are there: agrochemical based agriculture, organic agriculture, and genetically engineered crop based agriculture. Here, during 1950s, the whole plant could be regenerated from explants, that is any part of plant taken out and grown into a test tube under sterile conditions in specific nutrient media. This capacity to generate a whole plant from any cell or explant is called totipotency. Remember, this term will be asked. Next, coming to the nutrient medium, it must provide a type of carbon resource, that is sucrose, and also inorganic salt, vitamins, amino acids, and growth regulators like auxins, cytokinins, etc. And then here you see this method of producing thousands of plants through tissue culture is called micropropagation. Remember, producing thousands of plants through tissue culture is called micropropagation. Each of these plants will be genetically identical to their original plant from which they are grown, that is their soma clones. Soma clones, remember, means the plant that is genetically identical to their original plant or mother plant, that is called soma clones. Many important food plants like tomato, banana, apple have been produced on commercial scale using this method. Next, another important application of this method is to recover healthy plants from diseased plants. Even if the plant is infected with a virus, the meristem, mainly the apical and axillary, is free of viruses. Hence, one can remove the meristem and grow it in vitro to obtain virus-free plant. Scientists have successfully in culturing meristem of banana, sugarcane, and potato, etc. Next, scientists have even isolated single cells from plant, and after digesting their cell wall, have been able to isolate naked protoplast that is surrounded by plasma membrane. Isolated protoplast from two different varieties of plant, each having desired character, can be fused to get a hybrid protoplast, which can be further grown to form new plant, and these hybrids are called somatic hybrids. And this process is called somatic hybridization. When a protoplast of tomato is fused with the potato, and then they are grown to form a new hybrid plant combining tomato and potato characteristics. Then plants, bacteria, fungi, animals whose genes have been altered by manipulation are called genetically modified organisms. Genetically modifications has made crops more tolerant to abiotic stress, that is cold, drought, salt and heat, reduced reliance on chemical pesticides, that is pest resistance crop, help to reduce post-harvest losses, increase the efficiency of minerals using by plant. This prevents yearly exhaustion of fertility of the soil. Then enhance the nutritional value of food. Example, golden rice, that is vitamin A enriched rice. In addition to these uses, genetically modification has been used to create tailor-made plants. We already seen that in that PDF. Tailor-made means these are plants that are cultured and grown for specific purposes, such as high yield, disease resistance or improved nutrient content. These plants can be genetically modified to be pest resistant, drought resistant or herbicide resistant. That is called tailor-made plants. Then here the same example is here. You can see Bt toxin is produced by bacterium called Bacillus thuringiensis. In short, Bt is called. Bt toxin genes has been cloned from the bacteria and been expressed in plant to provide resistance to insect without the need of insecticides. In effect, creating a biopesticides. Examples are Bt cotton, Bt corn, rice, tomato, potato, soybeans, etc. First of all, Bt cotton. Here you can see some strains of Bacillus thuringiensis produced proteins that kill certain insects such as lepidopterans, that is tobacco boardworm, and armyworm. Questions are asked. Lepidopterans means tobacco boardworm and armyworm. Coleopteran means beetles. Dipterans means flies and mosquitoes. Bacillus thuringiensis forms protein crystals during a particular phase of their growth. These crystals contains a toxic insecticidal protein which kill insect. But Bt toxin protein exists as inactive protoxins. But once an insect ingests the inactive toxin, it converted into active form of toxin due to alkaline pH of the gut, which solubilize the crystals. Next, then the activated toxin binds to the surface of midgut epithelial cells, that is the small intestine of the particular insect, and create pores that causes cell swelling and lysis and eventually the causes of death of the insect. Then specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. The choice of genes depends upon the crop of the targeted pest, as the most Bt toxin are insect group specific. Insect group specific, remember. The toxin is coded by a gene that is XII, named XII. There are a number of them. For example, the proteins encoded by gene XI1AC and XI2AB controls cotton bollworm. That of XI1AB, it controls corn borer. Questions are asked, very important. Next, another example, pest resistance plant. Several nematodes, parasites, a wide variety of plants and animals including human beings. A nematode Meroidian incognitia infects the roots of tobacco plant and causes a great reduction in yield. A novel strategy was adopted to prevent these infections, that is based on the process of RNA interference, RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of specific mRNA due to the complementary double-stranded RNA molecule that binds to and prevents translation of mRNA. The sources of this complementary RNA could be from an infection by viruses having RNA genome or mobile genetic elements, that is called transposons, that replicate via an RNA intermediate. And then using agrobacterium vector, nematode specific genes were introduced into the host plant. The introduction of DNA was such that it produces both sense and antisense RNA in the host cell. These two RNAs being complementary to each other forms a double-stranded that initiates RNA interference and thus silences the specific mRNA of the nematode. Then here you see diagrams are there. Host plant generates double-stranded RNA, triggers production of against the nematode infestation. First diagram, roots of a typical control plant. Then second, transgenic plant roots 5 days after deliberate infections of nematode but protected through novel mechanism. Here you can see. Next in medicine. Let's see the PDF, what it says, applications in medicine. Here, recombinant DNA technology helps for the mass production of safe and more effective therapeutic drugs. The products from non-human sources induce unwanted immunological responses, but recombinant therapeutics does not have such problems. At present, about 30 recombinant therapeutics have been approved. Of these, 12 are being marketed in India. First, you can see where we apply this biotechnology in medicine, that is genetically engineered insulin, gene therapy, and molecular diagnosis. First of all, genetically engineered insulin. Insulin is used to manage adult onset diabetes. Insulin forms the pancreas of animals, cattle, and pigs, causes allergy or other type of reactions to the foreign protein. Now it is possible to produce human insulin using bacteria. See the diagram? Here you see human cell, chromosomes are there, and this particular gene is responsible for controlling the formation of insulin. This particular. So what we have to do, we slice up the chromosomes, and then again we took out from bacterium plasmid and isolated plasmid, and then plasmid cut with an enzyme. Here it is cut down like this, and that cut down plasmid is then joined with the particular gene of interest, you see. Then it transfer this plasmid, genetically inserted genes inserted in plasmid, then it transfer to particular bacteria, and the bacteria multiply and produce insulin. Then insulin consists of two short polypeptide chains, chain A and chain B. Here you can see chain A and chain B that are linked by disulfide bridges like this. These are the disulfide bridges, this one, okay, this one. In mammals, insulin synthesized as a prohormone. Remember, insulin synthesized as a prohormone. It needs processing to become mature and functional hormone. The prohormone contains an extra stretch called C-peptide like this. Here, this one is the C-peptide from here to this portion. Okay. So this is removed during maturation into insulin. Next, in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chain of human insulin. They were introduced in plasmids of E. coli to produce insulin chains. Chain A and B were combined by creating disulfide bonds to form human insulin. Then coming to gene therapy. It is a method to correct gene defect in a child or embryo. Here, genes are inserted into a person's cell and tissue to treat a hereditary disease. It compensates for the non-functional gene. First clinical gene therapy 1990 was given to a 4-year-old girl which have ADA deficiency, adenosine deaminase deficiency. It is caused due to deletion of the gene for adenosine deaminase. Treatment by bone marrow transplantation or enzyme replacement therapy is not completely curative. So what we do? We did the gene therapy for ADA deficiency, collect lymphocytes from the patient's blood and grow in culture. See the diagram. It is a patient and we have to collect the lymphocytes that are present in T cells. So we collect the T cells and grow in a culture like this. It is grown in a culture. Introduce a functional ADA cDNA. Introduce a functional ADA cDNA into lymphocyte using retroviral vector. You can see this one has to be introduced. Then they return to the patient. Then again it then goes to the patient's body. This should be periodically repeated as lymphocytes are not immortal. That's why it should be periodically repeated. If ADA gene from bone marrow cells is introduced into the host at early embryonic stage, then it could be a permanent cure. yearly embryonic stage. If it is detected, then from bone marrow cell it is introduced into the cell, then it could be permanent cure. Next, molecular diagnosis. Conventional methods, serum or urine analysis, are not suitable for yearly diagnosis of diseases. It is possible by techniques like recombinant DNA technology, PCR and ELISA. First of all, PCR. PCR stands for polymerase chain reaction. Presence of pathogen is normally suspected only based on symptoms. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of bacteria or viruses can be detected by amplification of their nucleic acid by PCR. Here, uses of the PCR to detect HIV in suspects, AIDS patients, to detect mutations in gene in cancer patients, and to identify many other genetical disorders. A single-stranded DNA or RNA tagged with a radioactive molecule is hybridized to its complementary DNA in a clone of cells. It is followed by detection using autoradiography. The clone having muted gene will not appear on the photographic film because the probe will not have complementary with the muted genes. Then what is ELISA? ELISA stands for Enzyme Linked Immunosorbent Assay. ELISA is based on the principles of antigen-antibody interactions. Infection by pathogen can be detected by the presence of antigens, that is proteins, glycoproteins, etc., or by detecting the antibodies synthesized against the pathogens. Here you see antibody, this is the binding site, target site. Next, transgenic animals. Before starting this, let's see the NCERT, what it says. NCERT, yeah. Same points are given. Here you can see at present about 30 recombinant therapeutics have been improved for human use and in world over, and in India 12 of these are presently been marketed. First, genetically engineered insulin. Here insulin can be orally administered to diabetic people or not. Why? Question will be asked, and here is the answer. Insulin cannot be orally administered to diabetic people because digestive juices in the stomach and intestine break down the insulin before it enters to the bloodstream. First point, insulin is a polypeptide chain, and when it reaches to the small intestine, it is absorbed as a single peptide and can no longer function like insulin. As you see, polypeptide chain, both A and B are present in a mature insulin protein, and A, B, C, all three are present in a pro-insulin protein. Remember. Next, protease in the digestive system would degrade the insulin due to its polypeptide nature, rendering it into inactive. That's why it is injected, not orally administered. Insulin used for diabetic was earlier extracted from pancreas of slaughtered cattle and pigs. Earlier, in older time, insulin from an animal source though caused some patient to develop allergy or other type of reactions to the foreign protein, because insulin consists of two short polypeptide chain, chain A and chain B, that are linked by a disulfide bridges. In mammals, including human, insulin is synthesized as a prohormone. Like proenzyme, it is a prohormone. It needs to be processed before it becomes fully mature and functional. And in a prohormone, there are A peptide, B peptide and C peptide chains are there. All three makes a proinsulin. And to make it a normal hormone, matured hormone, C peptide should be freed. The C peptide is not present in mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using rDNA technology was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of the human insulin and it introduced them into plasmid of E. coli to produce insulin chains. Chain A and chain B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. Next, coming to gene therapy. Here also, Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child or embryo. The first clinical gene therapy was given in 1990s to a 4-year-old girl with adenosine deaminase deficiency. This disorder caused due to the deletion of the gene for adenosine deaminase. In some children, AD deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to patients by injection. Then here, but the problem with both these approaches that are not completely curative. So at a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA is then introduced into the lymphocytes, which are subsequently returned to the patient. However, these cells are not immortal. The patient requires periodic infusion of such genetic engineered lymphocytes. However, if the gene isolated from bone marrow cells producing ADA is introduced into the cells at early embryonic stage, it could be a permanent cure. Then molecular diagnosis. Here you see recombinant DNA technology, polymerase chain reaction, and ELISA are some of the techniques that serve the purpose of early diagnosis. Here, presence of pathogen is normally suspected only when the pathogen has produced a disease symptom, and by that time, that concentration of the pathogen is already very high inside our body. However, very low concentration of bacteria or viruses at the time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acids by PCR. Here, PCR is now routinely used to detect HIV in suspected AIDS patients. It is being used to detect mutation in genes in suspected cancer patients too. A single strand of DNA or RNA tagged with radioactive molecule is allowed to hybridize into its complementary DNA in a clone of cells, followed by detection using autoradiography. And ELISA is based on the principles of antigen-antibody interactions. Now, transgenic animals. Let's see the PDF here. Transgenic animals. Here you see these are the animals whose genome has been altered by introduction of an extra gene by manipulation. Example, transgenic rats, rabbits, pigs, sheep, cow, and fish. Over 95% of the transgenic animals are mice. Benefits. What are the benefits of transgenic animals? Now, to study the regulation of genes on normal physiology and development. To study the contribution of genes in the development of a disease. Third, biological products. Fourth, vaccine safety testing. And fifth, chemical safety testing, that is toxicity testing. First, to study the regulation of genes on normal physiology and development. Example, study of complex factors such as insulin-like growth factor. Second, genes from the other species that altered formation of this factor are introduced and the biological effects are studied. This gives information about the biological role of the factor in the body. Next, to study the contribution of genes in the development of a disease, transgenic models helps to investigate new treatments for human diseases. Example, transgenic models for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Third, biological products. Transgenic animals are used to produce biological products by introducing genes which codes for a particular product. Example, human protein, alpha-1 antitrypsin used to treatment emphysema. Products for treatment of phenylketonuria, PKU, and cystic fibrosis. Here, emphysema, you can see it is the normal bronchioles and alveoli, and emphysema with dilation of the alveoli. See the diagrams. In 1997, Rosie, the first transgenic cow, produced human protein enriched milk, that is 2.4 gram per liter. It contains human alpha-lactobumin. It is nutritionally more balanced product for human babies than natural cow milk. And then vaccine safety testing. Transgenic mice are used to test the safety of the polio vaccine. It is reliable. They can replace the use of monkeys to test the safety of the vaccines. Then chemical safety testing, toxicity testing. Some transgenic animals carry genes which makes them more sensitive to toxic substances than non-transgenic animals. They are exposed to the toxic substances and the effects studied. It gives immediate results. Next is ethical issues. Let's see the NCERT what it says. Here the same points are there. You can see animals that have had their DNA manipulated to possess and express an extra gene are known as transgenic animals. And all over the world there are about 95% of existing transgenic animals are mice. First, the common reasons about that particular transgenic animals, that normal physiology and development, study of the disease, that is cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. Biological products, here you can see question are asked. There is a particular human protein alpha-1 antitrypsin used to treat emphysema, and similar attempt of being made for treatment of PKU, phenylketonuria and cystic fibrosis. 1997, first transgenic cow, Ruji, produced human protein enriched milk, that is 2.4 gram per liter. This milk contains human alpha-lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Then vaccine safety, chemical testings. Ethical issue. Let's see. I think it is the last topic of this chapter. Ethical issues. Here, problems of unpredicted results. Genetically modifications may cause unpredictable results when such organisms are introduced into ecosystem. Indian government has set up organization like GEAC, full form will be asked, Genetic Engineering Approval Committee, which make decisions about the validity of GM researches and the safety of the GM organisms for public services. Then it is the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned. Certain companies got patent for product and technologies that make use of the genetic material, plants, etc. that have been identified, developed and used by farmers and indigenous peoples of a country. Example, basmati rice, herbal medicine, turmeric, names, etc. Biopiracy. Basmati rice has a unique aroma and flavor. We all know India has 27 varieties of basmati rice. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati rice. This was actually derived from the Indian farmer's variety. Indian basmati was crossed with the semi-dwarf variety and claimed as a novelty. Other people selling basmati rice could be restricted by patent. Then, most of the industrialist nations are poor in the biodiversity and traditional knowledge. The developing and the underdeveloped world have rich biodiversity and traditional knowledge related to bioresources. It has to be developed laws to prevent unauthorized exploitation of bioresources and traditional knowledges. Indian Parliament has cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms, emergency provisions and research and development initiatives. Let's see the NCERT here. Full form GEAC, Genetically Engineered Approval Committee, which will make decisions regarding the validity of GM researches and the safety of introducing GM organisms for public service. Here, it is estimated that there are 2 lakhs of varieties of rice in India alone. And the diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor, and 27 documented varieties of basmati rice are grown in India. There is a reference to basmati rice in ancient texts, folklores and poetries as it has been grown for centuries. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati rice in the US and abroad. This new variety of basmati had actually been derived from Indian farmer varieties. Indian basmati was crossed with the semi-dwarf variety and claimed as an invention or a novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Even there are several attempts has been made regarding the products like turmeric, names, etc. Then coming to biopiracy, it is the term used to the refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payments. And Indian Parliament has recently cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiatives. And with this, we have completed this particular chapter with the easy one. So, thank you all to watch this video, and if you find this one is helpful, then give a like. Do not forget to comment, and new viewers, please subscribe my channel. Thank you. Jai Jagannath.